Thank you so much for the introduction. It's a great pleasure to talk this conference today. I'm Federica Michielin. I'm a postdoc in Paolo De Coppi's lab at UCL, Great Ormond Street Institute of Child Health. And today I'm going to talk about my recent study about the role of the confined environment in microfluidics that reveals a hidden contribution of self-organizing ECM during IPS hepatic differentiation. So as you all know, liver diseases are one of the leading causes of mortality worldwide, and congenital metabolic disorders contribute significantly to that. As animal models have often failed to fully recapitulate human liver functions, they have been replaced by human-induced pluripotent stem cells that allow to be differentiated into functional hepatocytes and therefore to study the mechanism underlying genetic disorders while maintaining the same genetic backgrounds of patients. And for this, both 2D and 3D organoid-based differentiation protocols have been developed. However, they still produce immature hepatocytes and sometimes they lack robustness, particularly due to donor-to-donor variability. And this is mainly because they are based on mouse liver development studies. So the in vitro differentiation protocols are based on the recapitulation of the in vivo development based on mouse studies through the sequential supplementation of exogenous factors that mimic key developmental stages, and particularly the paracrine effects between the mesenchyme and the developing hepatocytes in vivo. And through this supplementation of exogenous factors, it is possible to achieve functional mature hepatocytes through key stages such as definitive endoderm, hepatic endoderm, immature hepatocytes, and finally mature hepatocytes. However, endogenous cell-secreted factors have been also recognized to play a key role in developmental processes, but also in the maintenance of pluripotence, differentiation in general, and also in the reprogramming to iPSCs. And also the exposure to the exogenous signals that we provide through our differentiation protocols activates secretory activity, which can also influence the cell behavior despite the genetic programs of the cells. So in this study, we asked whether and to what extent is important the contribution of the secretome of developing hepatocytes to determining the hepatic fate. And to do that, we reasoned that this, differently from the standard Petri dish substrates, the microfluidic environment can greatly enhance the accumulation of cell-secreted factors thanks to the low culture volume to surface ratio, and therefore allowing to both identify these secreted factors, but also to study downstream effects. And we successfully demonstrated the efficiency of these platforms in two recent studies that we've published in our lab, where we demonstrated that microfluidics enhance both the differentiation of human pluripotent stem cells, but also the reprogramming of somatic cells to human-induced pluripotent stem cells. So taking advantages of these technologies, we adapted a previously developed protocol to differentiate human-induced pluripotent stem cells to mature hepatocytes by injecting fully committed endoderm-derived endoderm-committed iPSCs into microfluidic platforms, and then supplementing the exogenous factors proper of the differentiation protocols to finally achieve hepatocytes with homogeneous expression of adult hepatic markers such as albumin and NHF4 alpha, expression of cytochromes, which are key markers of hepatic differentiation, and functional activities in terms, for example, of secretion of alpha-1 antitrypsin to levels comparable to the one that can be obtained with primary human hepatocytes that to date are the gold standards of the hepatocyte cells. And then we developed this, we designed this study in which, in parallel, we differentiated definitive and uncommitted cells into mature hepatocytes, both in standard Petri dish that we define conventional culture condition, or in microfluidics. So cells are exposed to the same exogenous signals, but have different possibilities to accumulate cell-secreted signals during differentiation. We first analyzed the outcome through some RNA sequencing analysis, and we observed a full segregation from the iPSCs of the differentiated cells, and still a slight but significant segregation of hepatocytes derived in microfluidics compared to those derived in Petri dish. And we looked deeper into these differentially expressed genes, and what we found is that actually among the differentially expressed genes upregulated in cells derived in microfluidics, a high percentage of these genes belonged to genes associated with liver, according to the Human Protein Atlas database. Also, with some gene ontology analysis, we observed that pathway belonging to key metabolic process of the liver were enriched in cells derived in microfluidics. These results were only at the transcriptional level, so then we moved to some more functional analysis, and we decided to look at a unique property of hepatocytes, which is the detoxification of ammonia through the urea cycle process. And to do that, we labeled cells with ammonium chloride containing heavy nitrogen ions, and then we detected the urea secreted from the cells thanks to the labeling of the reagents. And what we notice is that fully committed hepatocytes derived in microfluidics actually display higher functional activities in terms of ammonia detoxification compared to those derived in Petri dish. So then we asked what is that in microfluidics contribute to a more mature phenotype. So we decided to analyze the secretome of cells during differentiation, taking advantage of a high-throughput proteomic technique based on the stable isotope labeling by amino acids in cell culture, or SILAC, coupled with liquid chromatography mass spectrometry-based analysis. And to do that, we labeled in different manners cells differentiating in standard Petri dish or in microfluidics. And specifically, we labeled cells in microfluidics with using amino acids containing heavy carbon and nitrogen ions. And then we collected supernatants during differentiation, and we collected also the lysates at the end of the commitment towards the hepatic fate. So this approach allowed us to measure the ratio of concentration of each protein identified by the mass spectrometry analysis. And therefore, it's a straightforward measurement of what is more accumulated in the microfluidics compared to the standard Petri dish. Also, this approach allows to discriminate between cell-secreted proteins and proteins already present in the media, or contaminants, for example. And of course, at the end, we looked at the proteins with an H12 ratio, meaning heavy to light ratio, higher than 1, which indicates higher accumulation in microfluidics. So, not surprisingly, we found that almost all the proteins detected with differential expression in microfluidics and in Petri dish were accumulated in microfluidics, given the ratio H12 higher than 1. So this is not surprising. However, when we did the gene ontology analysis of these proteins accumulated in microfluidics, surprisingly, we found that the first categories to be enriched was related to extracellular matrix proteins, both core extracellular matrix proteins actually, but also extracellular remodeling proteins. And we did a list of these proteins related to these categories, And we also did a sort of, we designed this functional network interaction diagrams, which is based on the functional connections among the proteins that we identified. And we did find, this is based on databases reported in the literature, that these proteins not only are accumulated, but they also have some functional connection among each other, meaning that probably they can have some downstream effects in terms of pathway activation or something similar. We also, to make sure that what we detected was exclusively related to the accumulation in the supernatants, we analyzed the lysates of this culture to make sure that we didn't observe any oversynthesis of these proteins. And indeed, when we analyzed the lysates through the same techniques, we didn't find any differences in terms of numbers of proteins accumulated in the two systems. However, proteins more expressed in microfluidics belong to categories related to metabolic processes, meaning that they are probably more differentiated comparing to cells differentiated in the Petri dish. And then we did some immunostaining analysis of some core extracellular matrix protein that actually were, that we actually could detect during human liver development, taking advantage of slices derived from 8 and 15 post-conceptional weeks of fetal livers. So these proteins were actually expressed at this stage. And in our systems, we found that in microfluidics, these proteins were not only more expressed, but also they displayed these sort of net-like structures, meaning that they were also probably remodeled in some way. So we can conclude that these proteins accumulated in microfluidics derives from the deposition of the accumulation of the soluble proteins that we identify to the SILAC technique. And then to further quantify this remodeling, we did some imaging analysis that allowed us to quantify the extent of the remodeling through the analysis of these elements that give an idea of how many structures we can observe through the remodeling of this deposition of these proteins. We also did some gene expression analysis because the deposition of extracellular matrix protein can actually be related to an activation of cell receptors. And indeed, we found an upregulation of integrin receptors. We also observed upregulation of the fibronectin gene itself, which was one of the main proteins identified, overexpression of epithelial markers, and most importantly of the c-Met receptor, which is the key receptor that allows the binding with the HGF factor, which is the main driver of hepatic differentiation. So this gene expression analysis also corresponded to a differential phenotype between hepatocytes derived in microfluidic compared to those derived in Petri dish, where we observed in the first a more epithelial defined phenotype, and also actually higher expression of mature hepatic markers, despite the very early stage. So we then thought that given this upregulation of integrin receptors and responsivity to extracellular matrix, cells derived in microfluidics were more prone to form aggregates and to further differentiate in a 3D environment, which we know it's key to further promote the maturation of hepatocytes. So we designed this experiment in which we we committed cells to the hepatic fate, and then we cultured cells in a 3D environment of matrigel drops, taking advantage of a self-renewing medium that has been used in the literature for the expansion of human primary hepatocytes. And also, we try to mimic the microfluidic environment by supplementing in a Petri dish, in a conventional culture system, the soluble extracellular matrix proteins that we found through the SILAC analysis. And we picked some proteins relevant to us, which was the collagen 1 and fibronectin, two core ECM proteins that we found in the analysis, and also SPARC, that was the first enriched proteins that we found, which is actually not well-known proteins, but it's involved in the remodeling of the extracellular matrix, and in particularly in the binding with collagen 1 and also fibronectin. So we first analyzed the formation of organoids, and what we found surprisingly is that, first of all, in microfluidics, with cells derived in microfluidics, we can obtain more organoids, and therefore more efficiently. But also cells treated with the soluble fibronectin can also give rise to a higher number of organoids. And similarly, even though not significantly, we obtain the same with collagen 1, not with SPARC. And we speculated that maybe SPARC alone is not very useful to this system, whereas maybe in the combination with other core extracellular matrix proteins can actually have a role. We didn't observe any differences in terms of organoids size, just the number of organoids, meaning that cells are more competent to form organoids. And also all of them display homogeneous expression of the hepatic markers. Therefore, confirm the hepatic fate. Then we moved forward and tried to differentiate these organoids towards mature hepatocyte cells, taking advantage of the factors involved in the functional differentiation of hepatocytes. And what we found, again, is that overall, with cells differentiated in microfluidics, we can obtain organoids with higher functional properties compared to those derived in Petri dish. And similarly, with cells treated with the fibronectin particularly. So in this case, we analyzed the ammonia detoxification, alpha-1 antitrypsin secretion, and albumin secretion. We also looked deeper at the phenotype of these organoids, and we picked the fibronectin-treated ones because we can, in this way, propose a protocol in which just the supplementation of soluble extracellular matrix proteins can actually improve the differentiation of hepatic organoids without using more complicated systems such as the microfluidics. And we did, what we obtained is the homogeneous expression of hepatic markers, but also, most importantly, we have evidences of 3D polarity. So the polarity of hepatocytes, it's very unique and very difficult to obtain in vitro, but we successfully obtained this, resembling what can be observed, for example, in liver tissue.
Federica, it's beautiful work, but we are running out on your conclusions. Fantastic. Thank you.
Yes, just to conclude that the confined environment boosts the hepatic differentiation, and this is because we think also the accumulation of extracellular matrix and extracellular matrix proteins are accumulated, which increase the deposition and the remodeling of the cells. This can also be replaced by the soluble supplementation of these proteins. And finally, we want to propose the development of this protocol for the direct and robust generation of functional hepatic organoids from hiPSCs. And sorry to be late. I want just to thank all my lab and all the people that contributed to the work. Thank you.